Pink snow, or watermelon snow, represents a community of organisms that include microalgae and bacteria. Western's own Dr. Codner is conducting a first ever large DNA survey with the newest technology in high throughput DNA sequencing. Using these new powerful environmental genomic techniques, they are starting to investigate questions about how these algae communities change with elevation and latitude, and how they may be affected by reduction of snowpack in the future.
I'm Robin Codner, and I'm an assistant professor in the biology department at Western Washington University. I work on algae, and I've been working on algae for 15 years, and I've done a lot of different kinds of things with algae. I currently mostly study marine microalgae, but last year I started a new project working on algae that live in snow. Algae is actually a grab bag term for a bunch of different things that do photosynthesis that aren't plants. It's interesting how all of these algae are related. They're actually pretty closely related and not that different. So algae as a group are super diverse, and you can find them in almost any environment. And so what I'm really interested in is also their evolutionary relationships. So the algae that live in snow and the green algae that you find on the beach, they're actually from the same group. If you ever see pink snow in the late spring or early summer, that is algae that are living in the snow, and they actually live in a community with other microbes, fungi, bacteria. I've been observing snow algae for years because I'm a mountaineer and I'm always finding myself up in the snow and have seen these communities, and I knew they were algae, and I was interested in who they are and what they were doing, sort of a passing interest. In recent years, the methods that I use to study algae communities, which are these really large DNA sequencing projects from the environment where we can identify all of the diversity from a single sample, those methods are really well suited for studying the snow algae populations, and no one has actually done that before with our new DNA sequencing methods. So it seemed like a really appropriate match for my interests. And so even though this algae that are on snow are pink colored, they're green algae as well. And they're just pink colored because they have a bunch of molecules in them that act as sunscreen that make them look pink. So I have a laboratory in the biology department, and so I have all the resources that I have in my lab as well as other resources in the biology department. So we have some really nice microscopes that I use, as well as instruments that help us amplify the DNA. So a lot of the samples are still in the queue for sequencing. So last year I was one of the principal investigators who received a grant from the National Science Foundation to build a molecular ecology facility at the Shannon Point Marine Labs, and that's down in Anacortes. And I work there, and for doing all of my marine work. But the part of that new facility that we built includes a new high throughput DNA sequencer. And so now we have our own sequencer here at Western. We've been in the process of setting that up this year. So hopefully all of these samples will be sequenced down at the Shannon Point campus. So we have all these samples being ready to be sequenced, and some of the delays with that have been setting up the new lab. But a lot of the other part of my research is doing the data analysis. So when you do really big DNA sequencing projects for the environment, you get about somewhere between 50 million and 300 million sequences per sample. And that's only become possible in the last couple of years. So it's really aided by advances in DNA sequencing technology. And because of that, to analyze our data, we need to use a lot of computers. And so some of the biggest advances that have happened in my lab in the last year have been on the computer side and on the bioinformatics side. So I have a number of undergraduates that work in my lab, and I also have 3 graduate students. And everyone is really excited about this project, and so we're going to have some group hikes this summer to collect snow algae. I have people aiding me in my research, so I developed a collaboration with the National Park Service this summer, and we integrated this project into their BioBlitz program. And the BioBlitz program is this outreach with visitors to the park so that they can participate in science. And so from there we've developed a citizen science component where I have these sampling kits that people can pick up at the ranger station in Marble Mount and then take them with them on their hiking trip. And if they see snow algae, they can scoop it into the tube, and the tube has a preservative for DNA so that I can pick the samples up later and then use them and process them for DNA and use them in my sequencing research. And then of course anybody who's interested can contact me to get sampling kits, and it would be great to have lots of folks. So you can find my information on the biology department website, which has my email and contact info and also a link to my lab website. The more the merrier, so I hope that we can get them in the hands of a bunch of Western students.
And I say this in that I think, so many different degrees that are important. And if I look at, whether it's in engineering or it's different STEM programs, I think so many of them are so important to what we've done at Skullcandy, what we've done at Nike. I think one of the things that's really special about having a liberal arts degree or having a history degree or an English degree is just, number one, so much is around how do you inspire people, and so much of that is around writing.